I'm your educator Sulagna Ghatak. Today, after DNA recombination, I'm gonna brief you about the topic DNA repair. So let's start. So first of all, DNA molecules, like all other biomolecules, can be damaged in numerous ways. And spontaneous damage due to replication errors, deamination, depurination and oxidation, etc. is compounded in real world by the additional effects of radiations and various environmental chemicals. Right. So most such damages in DNA are temporary because they are immediately corrected by a set of processes which are called DNA repair processes. So without DNA repair, a genome would not be able to maintain its essential cellular functions. So since repair systems must be capable of recognizing and dealing with each type of damage, then it is not surprising that there is a large number of different types of DNA repair system, isn't it? So DNA repair system can be divided into 4 types: direct repair, excision repair, double strand break repair and sauce repair. Sauce repair only occur in case of bacteria. So direct repair can be subdivided into 2 subtypes: photoreactivation and alkyl transferase. And excision repair can be subdivided into 3 types: mismatch repair system, nucleotide excision repair and base excision repair. And double strand break repair, as we have seen in case of recombination, that there are mainly 2 subtypes: homologous double strand break repair and non-homologous double strand break repair. Clear? So let's see in details. So in the process of photoreactivation, we can see that UV light is one of the major sources of damage to DNA chain and is also the most thoroughly studied form of DNA damage in terms of repair mechanisms. So the major type of damage induced by UV light is formation of pyrimidine dimers. That means dimer forms between the thymine or between the cytosine, in which adjacent pyrimidines of the same strand of DNA are joined by the formation of cyclobutane ring, resulting from the saturation of double strands between the carbon positions 5th and 6th. So the formation of such dimers can distort the structure of DNA chain and block the transcription and replication past the site of damage. So their repair is closely correlated with the ability of cells to survive UV irradiation. So the UV-induced cyclobutane pyrimidine photodimer can be repaired by the enzyme photolyase that has been found in bacteria and lower eukaryotes, but it can't be seen in case of human. Okay, and also the energy derived from the visible light. So here you can see that thymine photodimer have been found or recognized. Then by the help of photolyase enzyme and visible light, it become removed. Okay. Nextly, in the second type of direct repair system, it deals with the damage resulting from the reaction between alkylating agents and DNA. So the alkylating agents are reactive compounds that transfer methyl or ethyl groups to a DNA base, and thereby chemical modification can occur. A particularly important type of damage is methylation of O6 position of guanine, because the product O6 methyl guanine forms complementary base pair with thymine instead of cytosine. Okay, so the mutagenic lesion or damage can be repaired by O6 methyl guanine methyl transferase enzyme that transfers methyl group from O6 methyl guanine to a cysteine amino acid residue on its active site. Okay, so lastly, the potentially mutagenic chemical modification is thus removed by the cysteine residue, and then the original guanine is restored. Clear? Now, in case of mismatch repair system, it can detect mismatches that occur in DNA replication process in mainly 3 steps. First of all, it can recognize the mismatch base pair, then it determines which base pair in the mismatch is the incorrect one, and lastly it can excise the incorrect base and carry out repair synthesis. So there is a delay between post DNA replication and methylation processes of daughter strand, and this is the main window of opportunity that the repair system can scan daughter DNA polynucleotide for mismatches, and that makes the required corrections in unmethylated daughter strands. So the enzymes DAM or DNA adenine methylase and DCM or DNA cytosine methylase can identify and add convenient tags at 6th and 5th positions on adenine and cytosine respectively. So that then that can be detected by the other enzymes for the process repair. Clear? Now, in case of E. coli mismatch repair system, so this is the parent DNA strand and this is the daughter DNA strand, because parent DNA strand always contain or carry a methyl group. So, after the addition of the tag by the DAM and DCM enzymes on this position, mut-S can recognize the mismatch, and then it forms a complex with mut-L and mut-H. Then the mut-H endonuclease cleave the unmethylated DNA strand at GATC sequence, and then mut-S and mut-L together with exonuclease and helicase enzymes excise the DNA between the strand break and the mismatch, and lastly the gap becomes filled by the DNA polymerase and ligase. Clear? Now, in case of nucleotide excision repair, it is commonly known as UVR-ABC excinuclease system. So this is frequently called excinuclease because it refers its ability to directly excise an oligonucleotide. So in case of E. coli, this repair system is catalyzed by the products of mainly 3 genes, that means UVR-A, UVR-B and UVR-C. In human, basic steps are as similar as in E. coli, but the function are done by the different enzymes. So the function of UVR-A is done by the XPC. The function of UVR-B are done by the XPA, XPD and replication protein A or RPA, and the function of UVR-C are done by the XPF and XPG. Okay? Now let's see what is the process of nucleotide excision repair in case of E. coli. So first of all, UVR-A and UVR-B proteins come and scan and also recognize the damaged DNA by ATP hydrolysis. Then after scanning and recognizing, UVR-A leaves the UVR-AB complex. Then UVR-B melt the phosphodiester bond around the distortion, and then UVR-C come and form a complex with UVR-B. So if into the upper strand of DNA, this is the 5' end, then this will be the 3' end, and if this is the 5' end, this will be the 3' end. So this 5' end become cleaved by the UVR-C, and this 3' end can be cleaved by the UVR-B. Now the UVR-D releases the single strand 12 to 13 base paired fragment of oligonucleotide from the duplex, and at last resulting gap will be filled by the DNA polymerase 1 and sealed by the enzyme DNA ligase. Clear? Now, after nucleotide excision repair, in case of base excision repair, uracil can be formed in DNA by deamination of cytosine, which alters the normal pattern of complementary base pair, isn't it? Thus, it represents as a mutagenic event, and as uracil, hypoxanthine can formed from adenine by the process of deamination, and as well thymine also can form from 5-methylcytosine by the process of deamination. So this excision is catalyzed by the enzyme DNA glycolase, which cleaves the bond linking the base to the deoxyribose sugar of DNA backbone. This reaction yields a free nucleotide base and an apyrimidinic or apurinic site, or I can say AP site. Okay, so let's see in details the process. So in case of uracil excision repair process, uracil glycosylase hydrolyzes the glycosidic bond to release the uracil from DNA backbone to leave a free AP site. So this is the uracil base and this is the free AP site. That means this sugar contain no any base with it. Okay. Now the AP endonuclease cuts the DNA backbone at 5' position of the AP site. So this 5' position can be cleaved by the AP endonuclease and it leave a 3' OH end. And also AP exonuclease cleaves the DNA backbone at 3' position of AP site and leave a 5' phosphate end, and resulting gap at last become filled by the DNA polymerase 1 and ligase. Clear? Now in case of double strand break repair, as we have seen in my previous video, right, that there are mainly two types, homologous and non-homologous recombination, as homologous recombination occur mainly in case of prokaryotes and non-homologous recombination occur mainly in case of eukaryotes, right? So the double strand break repair can be repaired by the several homologous recombination mediated pathways. That means double strand break repair or DSBR and synthesis dependent strand annealing or SDSA. So the process is rec BCD cleave the 5' end of the from each strand. Then the strand invasion occur by the rec A, which is a mutation sensory protein, and one template strand form a nick, right? Then DSBR and SDSA form. Then in case of DSBR, holiday junction formation occur and branch migration occur by RUVA and RUVB proteins. Then RUVC do the resolution and at last gap filling occur by the DNA polymerase and ligase enzymes. Clear? Now the last repair mechanism is the sauce repair mechanism or save our soul. That means the principle here involved is that survival with mutations is better than no survival at all. That means it is an error-prone system because the strand contain incorrect base even though intact DNA strands are formed. So this is also known as bypass repair system because it allows DNA chain growth across damaged segment at the cost of fidelity or accuracy of replication. So the process is Lex A repressor or which is a promoter only allows the DNA strand to proceed into transcription when the all base pairs are perfect. Then the presence of rec A filament help to signal the cell to synthesize UMU-D and UMU-D1, Then UMU-C become required to form complex with UMU-D2. Then UMU-D2 prime C complex, which you have seen in the replication enzymes, right, which is also called DNA polymerase 5. So this complex of source gene permits DNA polymerase to proceed of past blocked lesion by forming a dimer with lex A repressor. Clear? So, hope your concept about this topic have been clear after this session. There has provided a link of e-quiz into the description box so you can score healthy marks and test yourself by those questions. And if you have any doubt or facing any problem during solving those questions, then after scoring you can ask me into the comment section, and also you can reach to us through our mail ID which is into the description box. I'm so much happy to say that in previous lessons I've got your lots of responses and doubts through mail, so don't miss the chance to get a healthy score. Lastly, thanks for watching and don't forget to subscribe our channel, then you can get notifications about my next videos which is coming on every Sunday. So stay safe, stay tuned. Bye.